Welcome, folks. In this video, we are going to focus on biology paper 2 for the year 2016, and we are dealing with a paper that was written by the internal candidates. Now, let's look at the first question. So, the first question, we have a detailed structure of an animal cell, and this cell, we are told, was seen under an electron microscope. So, an electron microscope is just a more advanced microscope compared to the light microscope. So this one will be able to show you some of the structures that are considered to be detailed. Now, the first question is identify the part labeled F and G. So our F is we can see how this structure is. So this structure is known as endoplasmic reticulum. Now, whenever there are these ribosomes on top or on the surface, then the name you add rough endoplasmic reticulum. So the rough has ribosomes on the surface, while the smooth does not have the ribosomes on the surface. So that's what you have to use whenever you are trying to differentiate the rough from the smooth endoplasmic reticulum. So part F is rough endoplasmic reticulum. Then part G. So part G, we are looking at this membrane outside here. So the part inside this membrane is a nucleus, and we label the nucleus by, we know that it's shaded. So this part that is shaded here is the nucleus. So what membrane that surrounds the nucleus? So the membrane that surrounds the nucleus is nuclear membrane. Okay, so now we move on to question B. Explain the functions of the parts labeled H and I. Then the total marks were 4. So we need to make sure that each part must have 2 marks. So how can we score 2 marks for each question? We'll have to look at the first one. We'll start by mentioning the part, then we give the function related to that particular part. So let's go back to our H. So what is H? So H, you can see that it's a sausage like structure, and this one is known as a mitochondria. So part H is mitochondria. Now, what is the function of mitochondria? Mitochondria can be in terms of the function, it can be given in so many ways. You can give mitochondria as the site where respiration occurs, or the site where energy is produced. So the key point here is just that mitochondria is responsible to produce the energy needed by the cell. So whatever the cell needs, that energy needed for the journey, for the work that should be done by the cell, is produced by this mitochondria. So if what we can say mitochondria is also known as a powerhouse. So that is the other name. In case you encounter a question of what is the function of a power, what is considered to be a powerhouse in a cell, so that is mitochondria. Then we'll say this is where respiration occurs. And if you look at respiration, respiration will give you the picture of how the foods that we eat. If you look at starch, how is starch or how is glucose broken down into water and carbon dioxide plus energy. So the whole respiration is about production of energy. So that's the name powerhouse. So it has to do with the production of energy. Then what else? So the other parts that we need is eye. So the eye is just the outer part here. So what is this outer part? This is a cell membrane. So what is the function of cell membrane? So cell membrane can also be answered in so many ways. It is a part that encloses the animal cell. Or the other function for cell membrane, we say it's selective. It is a selective membrane, meaning it allows some substances to enter and also substances that should be out. So therefore it acts as a security. It has to know, are these substances needed by the cell? Do we have substances that might affect the cell? So if so, then this part is the one that is responsible to allow what to enter and also what to leave the cell. So if the materials are not needed, then this party will act as a security. It will make sure that those substances that is not needed should never enter the cell. So say it allows some substances to go in and out of the cell. So here, by mentioning the part, 1 mark, the function, also 1 mark, we have the 2 marks. Also here, mitochondria, 1 mark. Then the function, we have gotten our 2 marks. So for questions like this, I advise you to first mention the part, even if the part is not required. Then question C, suggest 2 cell parts which would be present in figure 1.1 if it was a plant cell. Now remember, the cell that we are looking at here is an animal cell, and this question is asking us or maybe trying to tell us what are the parts that we think can be found in a plant cell but not in an animal cell. So the parts that you would expect to have for this cell, if it was a plant cell, then it's the cell, or we know that plant cells have the cell wall. Then the second one, we may have the chloroplast. So we may have the chloroplast. Now the cell wall, in terms of the function, the cell wall protects whatever that is inside the cell. Then also it gives that regularity shape of the plant cell. Then if you look at the chloroplast is just the site where the process of photosynthesis occurs, and process of photosynthesis, it is just the process by which plants manufacture their own food. So as you are revising and also studying, make sure you start with the notes so that you are guided on what to do and also what is needed. Then we have another question. Figure 2.1 shows the effect of pH on the rate of enzyme catalyzed reactions K and M. Now this is the enzymes, then in relation with digestion and also some region of the alimentary canal. So if you look at digestion, we are looking at the processing by which large molecules of food are broken down into simpler ones for two reasons: for easy absorption and also for easy transportation. Now, given the pH scale here, we know that from here, which is 0 up to 6 here, we are going to have an acidic medium. Then 7 will give us neutral. According to science, this is neutral. Then above 7, going to this side, we are going to have alkalinity or basic, which is a base. So we also have to look at, if you look at the alimentary canal that starts from the mouth up to the anus, so you just also have to look at which regions have the alkalinity medium and the acidic medium. Do we have the neutral medium if possible? So that will help you to answer questions. So now in the y-axis we have rate of reaction. So the y-axis is giving us how fast the reaction is proceeding. Then in the x-axis we have the pH concentration. So the pH is changing from the acidic to the neutral, then to the basic. So let's look at the question that is. So question A, what term is given to the pH at J and L? So at J, this is the highest point this reaction K reached. Also at L, it's the same. So this point is what we call optimum. So the word optimum is not only concerned with the biology. The word optimum is just the term that talks about the point, or the point at which something is at its best operation. Or you as a person, there are times where when you are studying, you feel like it's not making sense. Then there are times where you are studying, you are enjoying. There is a certain point that you reach, at that point where you feel like now whatever that you are studying is making sense and you are understanding that. So that point is what we call the optimum. So enzymes also have their optimum point. They have their point where they work the best. Now, beyond that point, things get bad. Now, in terms of the pH, here, we'll say optimum pH. Remember, it's not always optimum temperature. So what determines if the answer is optimum temperature or pH is, just look at whatever that you have in the x-axis. So in the x-axis, we are dealing with the pH. So that point is optimum pH, which is potential hydrogen. B, explain why the rate of reactions K and M goes down after point J and L respectively. So let's go back. So after reaching these 2 points, the two reactions respectively went down. Now let's see if we can analyze these reactions. So at this point here, the reaction is fast, or the speed of reaction is increasing at a normal rate or at the best rate. Then at this point here, the reaction is at its best. Now at its best, so maybe let's first go back to the numbers. Maybe we can see what was happening. Let me see if I can make one or two to understand this graph here. So let's look at the point. This is 0. So going this side, the pH of an acid is decreasing. Remember, the acidity increases when you go in this direction. So as the pH is decreasing, then the enzymes start to work well. As the pH is decreasing in the acidic medium. This enzyme is so at this 2 point something, maybe 2.5. At 2.5 is where the enzyme is now working at its best operation. Now, after this number 2 point something or 2.5, maybe at this point 2.7 somewhere here, we find that the reaction K started to slow down. Why? So it's because something happened to the enzymes. So at this point they were working at their best point. Then after that the enzymes became denatured. So what is to denature? What does it mean to denature? So to denature simply means the enzymes have lost their original shape. Remember, one of the characteristics of the enzymes, we say enzymes have specific what? Shape. Like for example, lock and key. We may have an enzyme with this shape. Then the food substrate must be like this so that it has to fit here to make the enzyme substrate complex. Now, if this is our enzyme, after being denatured, this enzyme may form the shape something like maybe something like this. Now the question is, can this substrate fit here? No. So after the denaturing, the enzymes cannot now work. It's not like maybe they are used up or maybe they have gone somewhere. No. Whenever you look at denaturing, whether it is in connection with temperature or pH, so whenever you look at denaturing, just have it in mind that the enzymes have changed their original shape. As the result, they cannot fit with their respective substrate. So that is what now caused these two reactions to started to slow down. So the enzymes were denatured. So the reactions K and M reached their optimum pH, and beyond that the enzymes were denatured. Okay, so the enzymes were denatured. As a result, the two reactions went down. Okay, so now let's move on to question C. Name the regions of the alimentary canal where reactions K and M take place. So let's go back. So K is in acidic medium because of the numbers. M is in basic medium or alkalinity medium. Okay, so let's go back. K, acidic. Which region of the alimentary canal has the acidic medium? So that is the stomach. We know that in the stomach we have hydrochloric acid. So that hydrochloric acid produces or it makes the medium to be in a acidic condition to favor the enzymes that operates there. Then M, alkalinity, can be the mouth. We know that the digestion starts in the mouth. In the mouth we have the basic or duodenum also, or we can just say small intestine. So these two regions also have that. Then D1, suggest one enzyme which catalyze reaction M. So M, we are saying it's where the basic is. So this can be salivary amylase, if you choose the answer to be mouth. Then if it's the duodenum, it can be pancreatic amylase. Okay. Then step 1 food nutrient which can be catalyzed in reaction K. So reaction K is the stomach. So the type of digestion that occurs in the stomach is for proteins. State 2 factors other than the pH which affect enzyme activity. So the first one, temperature. Then the second one, enzyme concentration. Also even the food substrate concentration can also be the answer here. Okay, so now let's go to third question. So third question, figure 3.1 shows a cross section through a stem of a plant which had been previously dipped in a red dye for 6 hours. Okay, so there are parts here, and this is according to the arrangement of these parts that are visible. We can tell that this is a dicot stem. So now we have structure N, P, O, Q, as well as R. Okay, so now what's the first question? A1, identify the parts labeled N. So N, this part here. So this is cambium. Okay, then P. P, we have the phloem. Then Q, xylem. Now, you need also to look at maybe something that can help you to differentiate the two in terms of location, phloem and xylem. So remember, xylem is found inside, phloem is found outside. So maybe you can use the alphabetical order. We know that xylem comes comes after P. So P, phloem is outside. Then as you enter inside, you have X. Maybe that can help, since at times during examination, these are simple things that sometimes may confuse you. You may be able to remember the parts, but you struggle. Which one is the xylem? Which one is the phloem? So the xylem is inside, X inside, P outside. I don't know if that can help. Then which labeled part in figure 3.1 would be stained red? Okay, so we are told if this was dipped in a red dye for 6 hours. So the question is indirectly asking us which vessel will transport the red dye. So let's look at the red dye. Red dye is a solution. So which party would transport solution? We know that solution is transported by a xylem, so it's part Q. Give a reason for your answer in the A2 above. Xylem is a vessel which transports solution. So since it is the one that is transporting the solution, that part will be the same, or it will be the part that will be stained red, that will show that color. Then B1 suggests 3 factors that would increase the rate of movement of the dye in the stem when it is dipped in the red dye. So the first one, high temperature. So as the solution will try to evaporate at the high rate, hence the high movements. Also high light intensity. Then also low humidity. So humidity is just considered as the amount of water vapor in the atmosphere. Excuse me. So if the humidity is low, the rate of transpiration will be high. As a result, we are going to have the process to be at the highest movement. Then to name the process that will cause the movement of the red dye in the stem. So this is the transpiration pool. So it is transpiration pool. Okay, so now let's move on to question 4. Figure 4.1 shows a certain type of flower. Okay. Then we have part S, T, U. Okay. Then the question is identify the parts labeled S. So what's our S? So these are anthers. The function of anthers are there to produce pollen grains. Then what's our T? T is stigma. Stigma is the female part that receives the pollen grains. Then U, ovary. So ovary contains the ovules, and that is where the double fertilization occurs. Then B1, identify the type of pollination that occurs in the flower. So for this flower, you can see these, they are down here, then these. Okay, so the type of pollination that we have here is wind pollination. Pollination, we are just looking at the transfer of pollen grains from the anthers to the stigma. Now, give 2 features from figure 4.1 which support your answer in B1 above. Okay, so we can see that we have long filament. These filaments are long. Then not only that, the anthers are above this. So if the wind were to blow this, if it shakes, if this shakes, then these will be able to fall on this. So that's a picture here. Then also we can see that we have feathery. So feathery. So the first one will say long filament. The second one, the anthers are above the stigma. Okay. Then C1, step 1, other type of pollination other than the one mentioned in one above. So this is just insect pollination. So here is just the process where we are looking at for pollen grains to be transferred from one flower to another, or from the anthers to the stigma. The insects are involved, so the insects are acting as the agents of pollination. Now give two characteristics of the flower where the type of pollination mentioned in C1 above occurs. So we can say that they have bright petals to attract insect. Then the second one, they have a nectar fluid. So a nectar fluid is just a fluid that is sweet. So because of that sweetness, the insect now may visit this. As the result, we have the transferring of pollen grains. Now we look at question 5. Figure 5.1 below shows pedigree diagrams for two families A and B, outlining inheritance of a sex-linked disease called hemophilia. So we have family A and family B. Now from the family A, we can see that there was a son who was hemophiliac. Then in family B, we don't have a child who is hemophiliac. Now let's have a look at the first question. A1 from figure 5.1, which family has a parent who is a carrier? So we know that it's family A. Why do we say so? Because there was a son who was hemophiliac. So under pedigree, whether given a key or not, whenever the box is shaded, that individual is sick. If it's not shaded, then he is okay. Now B1, using the symbols JH, XH capital letter and XH small letter, state the genotypes for offspring J and H if J is a carrier for hemophilia. So let's look at HH. We know it's shaded, so that one, our H is a male is XY, and whenever a male is sick, we just have to put one small letter H. Remember, the Y does not contain these sex-linked characteristics. Then J, we are told J is a carrier, so we need to have one capital letter and one small letter. Now, using the genetic diagram, show whether the offspring would be hemophiliac or normal when H and J are crossed. So first, begin by, let's look at parental phenotype. So parental phenotype, so say the husband is H. So the husband is H, the wife is J. So H will say hemophilia, which is hemophiliac, because that person is sick. Then the wife, carrier. Then let's look at the parental genotype. So for the husband, we know it's this. Okay, then what do you do? So let's look at the gametes. Gametes of P1, which is parental generation. So you just have to spread this. Then let's look at now random fertilization. So this is a chance of having a daughter who is a carrier. We have a son, not a son, a daughter who is sick, small letters. Whenever we have two small letters under XX, that individual is sick. Then here we are going to have a son who is 100% okay. Here, a son who is sick. So say genotype. Genotype of F1. So this is how we can answer this question. So that's it for this video. Thanks for watching.